Hello, darling students. This is me and Mr. Theodore saying hello and that we hope you are doing well today. Get ready to tuck in, maybe wear some comfy pants, get your notes out, specifically that PowerPoint, and we are going to start talking about the heart. All right, we are going to start with the functions of the heart, and specifically what we need to talk about first is going to specifically be this little separation here between the pulmonary and the systemic circulation that is going to be caused due to the heart. Technically. So do pay attention to this sort of right side, left side, what is happening. So the right side of the heart is going to, receive blood from the entire body. Keep in mind this is going to be low oxygen blood that it's going to receive, and it is then going to pump that low oxygen blood into the pulmonary circulation, which, remember, this is going to be your lungs. So it does say that here, but just to emphasize, that blood, which is going to be, and this is going to be a little messy, high in oxygen, is going to return to the left side of the heart. The left side of the heart is then going to pump that nice high oxygenated blood through the systemic circulatory system, which is going to essentially just be the rest of the tissues of your body. And then again, we're just going to keep in that nice cyclical kind of pattern, and blood is going to then return to the right side of the heart from the body. So that is sort of an important little kind of how everything's going about, but then specifically getting into those functions of the heart, it is going to generate blood pressure. This is going to be due to the contractions of the heart. So kind of make a little note there that that is caused by contractions of the heart. The second function is going to be routing blood, meaning it is just going to separate between those two systems. We were just talking about the pulmonary and systemic circulation systems. The third is going to be ensure a one-way blood flow. This is going to be through valves. We wouldn't want any backflow. That would cause some issues. So it is going to be a one-direction flow. And then the fourth function is going to be regulating blood supply. And you can make a little note here that what this means is it's going to be a supply and demand of oxygen, nutrients, whatever your tissues are needing. Your heart is going to ensure that it is going to get what it needs. So if you need to be taking in more oxygen, your heart might start pumping a little bit faster in order to circulate that blood into your lungs, pick up the oxygen, drop off the CO2, and then get that oxygen to your tissues. So this is a nice graphic showing the circulatory system kind of split into those two parts, the systemic and the pulmonary circulation. You can see that the pulmonary circulation is going to be that pathway of blood from the heart to the lungs and then back, while the systemic is going to be through the entire body. All right, so talking about size, shape, and location of the heart. If you wanted to, you could make a little fist with your hand. If you put your fist right against your manubrium, your sternum, and just go slightly to the left, that is typically where your heart is going to be. And again, it's kind of roughly the size of a closed fist, and specifically to each person, your body should be in general evenly sized. So if you're kind of a larger person, chances are your heart's going to be a little bit on the larger side. You're probably going to have larger hands, etc. So there are various parts of your heart that I want you to know. These terms should be relatively familiar and kind of self-explanatory. The apex is going to be that bottom portion of the heart that is going to have that blunt, rounded point, kind of a cone. So if we look at our diagram over here, it's going to be this guy down here, that little point down there. while the base is going to be this top portion. So it's going to be the flatter part where we have a lot of vessels coming in and out. Back to the location of our heart, it's going to be located within the thoracic cavity, but within a kind of sub-cavity called the mediastinum. Mediastinum, words sometimes, you know how I get. But knowing this area is very important for the purpose of performing CPR, or cardiopulmonary resuscitation. So obviously if you don't know where the heart is supposed to be and you just start hammering down on someone's chest and you're right above their sternum, you're probably going to break their sternum and probably cause more problems than good if you are not in the correct part of the body. So it is very important to know where the mediastinum and thus the heart is within the thoracic cavity. All right, getting into a little bit of the anatomy of the heart, and I'm not going to go into this in extreme detail because a lot of this we're going to cover in lab, but we are going to cover this briefly here. So the pericardium is going to be a sac that is going to surround the heart. It is split into two layers, the fibrous pericardium and the serous pericardium, and they kind of are describing themselves in their own title. The fibrous pericardium being obviously fibrous. It is the outer layer. It is going to prevent over distension, so preventing the heart from distending out too far. So it's going to kind of act as, I don't want to say like a cage, but it is ensuring that the heart does not overstep its bounds, if you will. It is also going to act as an anchor within the mediastinum, for the heart that is. That serous pericardium is going to be a thin, transparent inner layer made of simple squamous epithelium and is divided into two parts, the parietal pericardium and the visceral pericardium, or the epicardium, which is going to be touching the heart's surface. These are very important in that they are going to have a pericardial cavity between the pericardium and the heart, which is going to be filled with pericardial fluid. Make a little note underneath pericardial fluid that this is going to act as a kind of lubricant as the heart is beating. We don't want to have very much friction going on because it needs to continuously beat, obviously. That is going to potentially cause inflammation if it becomes irritated in some capacity, but this pericardial fluid is going to reduce that risk and act as a lubricant as the heart is beating. All right, so let's talk about the layers of the heart wall. We have first the epicardium, which is going to be that serous membrane on the outside. It's going to be a smooth outer surface. The myocardium, which, remember, if you ever see this term myo, and a lot of you got this right in lab, it's going to mean muscle, right? So myocardium, muscle. So that is the heart muscle layer, if you will, made of cardiac muscle tissue, and it is responsible for causing the heart to contract. And then the inner layer is going to be the endocardium, endo meaning, you know, like inside, right? That is going to be a smooth inner surface of all the heart's chambers. And you can see these trabeculae carnae that make up this inner layer here, right? And this is going to be incredibly smooth, especially on the inside walls of the ventricles. We are wanting this to be nice and smooth in order to not cause too much, again, friction between blood and the inner layers of the heart. We don't want that blood to be lingering in any of our chambers. We want it to slide along nice and easy. And then the last thing that I didn't mention on the slide, I do apologize, is the pectinate muscles. These are going to be muscular ridges in the auricles. Remember, the auricles are going to be those fatty little ears, is what I call them, on the outside of the heart. And if I could gently kind of circle their general location here, those are the auricles there on the heart. And we'll talk about that a little bit more here in just a second. All right, moving on into these primary chambers within the heart, we have the atria and the ventricles. The atria are going to receive blood from outside of the heart, while the ventricles are going to receive blood from the atria. So kind of make a little note of that the atria are going to supply blood to the ventricles, but outside of the heart is going to be supplying blood to the atria. On the outside of the heart, as well as kind of attached to the atria, are going to be those little fatty ears, the auricles, that I was just talking about. The purpose of our auricles is going to be to increase that chamber, so specifically the atria. It's going to allow the atria to expand further. So how blood enters the heart is going to be through a few different vessels. You have the superior and inferior vena cavas, and then from our pulmonary veins. And when we go over the diagram itself, we're going to kind of look at the flow of blood. So just for now, just know that these vessels are going to be receiving blood from the outside of the heart. This is how blood gets into the heart. How blood leaves is going to be from our ventricles through the pulmonary trunk or the aorta. Those are the only options of how blood is going to leave the heart. And specifically, it will go from ventricle to pulmonary trunk or from ventricle to aorta. We do have a few surface features that you need to know. Specifically grooves, such as the coronary sulcus and the anterior and posterior interventricular sulci. And we'll look at those a little bit more as we look at pictures. But do keep in mind that this is going to be locations where we are going to have coronary vessels. So those arteries and veins that are going to be supplying the heart with blood or taking away waste. All right, so moving on into the surface of the heart with this little diagram here. So we've got a lot of vessels going on with different colors. I do want to make sure that you understand the difference between what these colors are showing you. The blue vessels are going to indicate where you have vessels that are carrying low oxygen blood, while the red is going to be highly oxygenated blood. So kind of keep that in mind. A lot of people think that blue means veins and red means arteries. That's pretty common across the board, what people kind of have an understanding of, and in general that can help you a lot, especially when it comes to the veins and arteries and arterioles and venules when we get further along. But closer to the heart, that doesn't always mean that it's an artery and a vein, depending on the color. Just know that it's low oxygen or high oxygen is specifically what it's showing you. So let's go over a few of these external structures here. So first and foremost, you're going to have this big guy coming in right here, and then this big guy right here. So that is the superior and the inferior vena cava. Both of those are going to empty into this structure here, which is the right atrium. So keep in mind that this side is going to be right and this side is going to be left, because we are looking at this heart as if it were in someone else's body in front of us. So we have our right atrium. It is then this blood that we are bringing in going to pass through a valve, and we'll talk about that valve here in a minute, into another chamber, this guy down here, which is the right ventricle. From the right ventricle, it's going to go up and out through another valve into the pulmonary trunk. So the pulmonary trunk is going to be an artery. Keep that in mind. I always like to tell students arteries are going to carry blood away from the heart, so away, and veins are going to carry blood toward the heart, so away. So we are going to then split, and that is going to turn into our pulmonary arteries, where we are then going to enter into the lungs, get nice and oxygenated, and that blood is going to come back through these vessels on the right and left, called the right and left pulmonary veins. So bringing blood back in. This is going to empty into the left atrium, where we are going to pass through a valve and enter into the left ventricle. And from there, we're going to kind of pass underneath here and then up and out through the aorta. Here's a nice, lovely, real heart. I think this is a fantastic picture. It is a good one to study from. This is a beautiful heart. So kind of just make a note, everything we talked about in that last slide is on this slide as well. Well, for the most part. All right, so this is the surface of the heart, but from the back. You can see a lot of those main structures that we were talking about before, but if you're a little confused, just to give you a little orientation, this is now the left side and this is the right side because we're looking at the back of it, right? So we are going to have the right atrium, the right ventricle, the left atrium is now up and over there, and then the left ventricle is down here. Keep in mind that these big structures on the back here are going to be the superior and inferior vena cavas entering into the right atrium, and then we're going to have this structure right here, which is our pulmonary arteries. So if we were seeing this from the front, we would be able to see that pulmonary trunk, but because it's from the back, obviously we cannot. We can see our pulmonary veins entering into, I'm so sorry, our left atrium. I should have included more back here, my apologies. And then we can see a little bit of our aorta coming up here. All right, so here is a slide discussing coronary circulation, specifically the arteries. So these are going to be the vessels that are going to be supplying blood to the heart itself. Do keep in mind that the blood that is going to be supplied to the heart is going to be directly off of the aorta. And the aorta is going to be containing blood that is the highest oxygenated blood in your entire body at any given point in time, unless there's something wrong, of course. So that is incredibly high oxygenated blood, which makes sense. We're going to supply it directly off of the tap there to the heart, because that's going to be a muscle that is constantly working for you, needing a lot of oxygen and producing a lot of waste. So we have the left coronary artery, which is going to exit the aorta near the coronary branches. We have the anterior interventricular artery. This is going to be in the anterior interventricular sulcus, which is going to be that little depression going down the middle of the heart on the front. You have the left marginal artery, which is going to supply the lateral wall of the left ventricle with highly oxygenated blood. And then you have the circumflex artery, which is going to extend to the posterior aspect of the heart. Next, we have the right coronary artery, which is going to exit the aorta just superior to the point where the aorta exits the heart. So again, kind of right off of the tap, the highest oxygenated blood that you can have in your body. It is going to lie within the coronary sulcus. Again, that little depression there, and I believe the next picture or the next slide is going to show us a picture of all of this. It is going to be smaller than left, and it extends to the posterior aspect of the heart. From there you have the right marginal artery, which is going to be lateral to the lateral wall of the right ventricle, and the posterior interventricular artery is going to lie in the posterior interventricular sulcus. Again, anytime you see that word sulcus, I don't know that I said this before, that just means a depression. A sulcus, I always think sulcus, sunken depression. But that is going to supply the posterior and inferior aspects of the heart with that highly oxygenated blood. And yes, here we go. Here are all of those arteries that we were just talking about, kind of labeled out. Pause this if you'd like to, if you want to kind of focus on this for like a hot minute, but I don't feel the need to kind of go over exactly what I just went over when this just shows you where everything is. So give it a little pause if you need to take a peek at this a little longer. Moving on to the coronary circulation of veins. So you have the great cardiac vein, and this is going to be kind of a bigger guy here, which you can see it if I can get my little pointer going right here. So we've got our great cardiac vein there. And then it's going to have a small cardiac vein that is going to drain the right margin of the heart. And then the coronary sinus, which is going to be veins that empty here and then into the right atrium, so directly back into the heart. All right, so moving on to our heart chambers and kind of associated structures with them. We have talked about them a little bit before, but there are a few things that you do need to know about each one. Let me change my color here. So first we're going to start with the right atrium. It's going to have three major openings to it that again all receive blood returning from the body. So kind of make a little note of that. The right atrium is going to receive blood from the whole body. And remember, this is going to be low oxygen. And then those structures, we've already talked about all of them, the superior vena cava, the inferior vena cava, and then what we were just talking about, that coronary sinus. The left atrium is going to have four openings, and they are all pulmonary veins. And do keep in mind that this is going to be highly oxygenated blood that is coming into that atrium. There is a structure between our atria, which is going to be the interatrial, try saying that 5 times fast, septum. So keep in mind, anytime you see this word septum, it's just meaning some type of structure that is separating one chamber from the other. So whether this is like, you can kind of think of your nasal septum, so it's separating your nostrils, but in this case it is going to be separating our two atria. It is going to contain a depression, or kind of a sulcus, which is going to be this fossa ovalis. This is a remnant of a fetal shunt. And a shunt is just going to be kind of something that allows something else to drain into another thing, if you will. It is called the foramen ovale, which, if you remember from the first part of A&P, foramen is just going to mean that it is a hole. So this is going to be a hole between your atria when you are a fetus. As you become not a fetus, so an infant or a newborn to an infant to a toddler, and thus into, you know, a small person, etc., that is going to close up. That's very important for it to close up because you are not going to need it anymore. If it does not close this hole, that's going to cause problems as an adult because you will be leaking blood that is low oxygen to high oxygen, and thus you will not be getting enough oxygen to your tissues. So it is very important for this to close up and create that little depression called the fossa ovalis. Moving on to our ventricles, you have something called the atrioventricular canals. These are going to be openings between the atria and their respective ventricles. If you were in attendance of lab, then potentially you know that this area is going to contain valves. So kind of keep that in mind. So the right ventricle, let's see if I've got any colors I haven't used. Hello. The right ventricle is going to open up into the pulmonary trunk and then thus go to the lung. So sorry about that, guys. I kind of got cut off on that last slide, and instead of re-recording all of it, I'm just going to kind of continue where I feel like it left off. So I've got the right ventricle here, which is going to open up into the pulmonary trunk. Keep in mind that that is going to be an artery that is taking our blood to our lungs for gas exchange to occur. From there, we're going to have our blood come back and thus go to the left atria and then into the left ventricle here, which is then going to open up into the aorta and then out through the entire body. From there, the last structure that we have that is associated with ventricles is going to be the interventricular septum. Just like the interatrial septum, this is just a wall that's going to separate our two ventricles. All right, so let's talk about the flow of blood through the heart, because I think this is the best way to learn the different parts of the heart, including the valves. So real quick, let's talk about these valves. So you are going to have two AV valves. So these are atrioventricular valves. So it is between the atrium and the ventricle, and that is just going to be this lovely little valve here, specifically between the right atrium and the right ventricle is going to be the tricuspid valve. So blood is going to come in through the superior and the inferior vena cava into the right atrium. From the right atrium, we're going to pass through this tricuspid valve and into the right ventricle. From the right ventricle, we're going to go up into the pulmonary trunk, but not before we pass through this other valve, which is known as the semilunar valves. That is the pulmonary semilunar valve, which is this first guy here. So aptly named because it is going to be going to the pulmonary trunk and thus into the pulmonary arteries and out of the heart, where it is then going to go to the lungs, become oxygenated, come back in through the right and, well, left and right pulmonary veins, which are going to enter into this left atrium, passing through another AV valve, which is going to be our bicuspid or mitral valve, and into the left ventricle. From the left ventricle, we're going to go up and underneath here, pass through another semilunar valve, which is then going to be the aortic semilunar valve, which is a signal that we are entering into the aorta. And that is, in general, with all the valves, how we are going to move through the heart. Now, this next slide is going to talk about those valves kind of more specifically, but now you know where they are. So, as I said on that last slide, the AV valves, the atrioventricular valves, are going to be the valves that are going to be between the atria and the ventricles. Now, what they kind of look like is, I'm gonna very poorly draw this. So this is going to be, in general, our little valve here. the surface of our heart, specifically in the ventricles, is going to have these little projections coming up like that. I'm going to kind of color in our valve just so we can kind of keep it distinguished from the other parts. Now, what's kind of interesting about our AV valves is that it's going to have these little stringy bits coming off of the leaf-like cusps, and they're going to attach to these projections from the heart wall. So these projections are the papillary muscles. So these are special little muscles that are going to relax and contract and pull on these strings that I just drew out here. Those strings are called chordae tendineae, and these chordae tendineae are going to become tighter or relax depending on whether or not these papillary muscles are going to contract. So what gives our valves their names? Because you notice that they had two different names for the right and left side. So the right is going to have 3 cusps that make up the valve, and that is going to be known as the tricuspid valve. Makes sense. While the left is only going to have 2 cusps and thus be the bicuspid. So the one that I drew up there is going to be that first valve, our tricuspid, while the other one is the bicuspid. You can also call it the mitral valve, but I think bicuspid makes more sense. I always like to tell students, because we're going from the right side of the heart to the left side, if we follow the flow of blood, we are going to try before you buy, if that makes sense and kind of helps you remember these things. The other valves are these semilunar valves, the pulmonary and the atrial or aortic valve. Not sure why it says atrial. I'd call it the aortic. I think that is incorrect there. Ignore that. It is the pulmonary and aortic valve, and these are going to have each cusp shaped like a cup or a crescent, and when the cusps are filled, the valve is going to be closed, but when the cusps are empty, that is when that valve is going to open, as opposed to by the chordae tendineae having tension or no tension, which is going to allow those valves to snap shut, such as with the AV valves. I skipped through a few of the last slides, so don't worry, you didn't miss any of the audio with that. In fact, you might have even heard me breathing through them. If you want to go back and pause them and take a peek at them a little bit further, you may, but I feel like I've gone over this a few times actually, and if you need to, you can always go back to what I was lecturing before about the flow of blood through the heart, as well as anything you may have missed about the valves. I just felt like these pictures are very useful, but I don't need to harp on everything a lot since you have the ability to kind of go back. So this is the flow of blood through the heart. The only thing that I would potentially add in here, well, this is just through the flow of blood through the heart. I'll get to how I like the flow of blood through the whole body here in just a minute. So just give me a second. It might be at the very end of this lecture, but we will get to that, I promise. But this is the flow of blood specifically through the heart. And it is showing you where you're going to have low oxygen blood, gas exchange, and then high oxygenated blood with that red and blue. Now that we have talked about the flow of blood through the various anatomical structures of the heart, let's talk a little bit more about the histology of the heart. This is, if you remember histology, going to include the tissues of the heart, if you will. So the heart skeleton is going to consist of a plate of fibrous connective tissue between our chambers, so between the atria and the ventricles. It is going to have fibrous rings around the valves in order to help support those valves and, of course, keep them in place. And it is going to serve as electrical insulation between the atria and the ventricles. And when we talk about how the heart stays coordinated and how it pumps, that will make a little bit more sense. So just kind of put a little pin in it for just a moment. And then this heart skeleton is also going to provide a site for muscle attachment. So you got to have kind of a basis to attach your muscles. Just literally think of how the skeletal system and the muscular system go hand in hand, that your bones act as leverage. This is the exact same concept. All right, moving into specifically cardiac muscle tissue. So if you were able to see some cardiac muscle tissue from the first part of A&P, you know that it looks very similar to skeletal muscle tissue, but there are some big differences. So cardiac muscle tissue is going to be nice and elongated. It is going to be branching, and the cells are going to contain 1 to 2 centrally located nuclei, so they are multinucleated. It is going to contain actin and myosin myofilaments, so just like skeletal muscle tissue, as well as striations and a sarcoplasmic reticulum. The big important difference between cardiac muscle tissue and skeletal muscle tissue, aside from a few things, but I consider it one of the big ones that tells you what you're looking at, is the intercalated discs. These are going to be specialized structures that help with cell-to-cell communication. These cell membranes are going to be interdigitate, and you're going to contain desmosomes, which are going to hold our cells together, and there are going to be gap junctions, which allow action potentials to move from one cell to the next. Specifically with cardiac muscle tissue, it is going to be electrically Electrically, cardiac muscles of the atria and the ventricles are going to behave as a single unit. There we go. So we are going to coordinate the way that the chambers of the heart function together. They do need to function kind of like a well-coordinated ballet, if you will. Otherwise, if one contracts or relaxes at the wrong time, as one of its partnered chambers is contracting and relaxing and everything's kind of off beat, if you will, we're going to have a lot of issues. Blood is not going to properly move through the heart, and thus you are not going to be able to oxygenate your tissues properly. So in order to keep everything moving smoothly, we are going to use electrical signals, and we'll talk about that here in just a moment. All right, moving into the conducting system. This is going to be where we relay action potentials through the heart and kind of how we do that. What this is composed of is going to be modified cardiac muscle cells that form 2 nodes, which you can kind of think of as kind of knots or lumps, if you will, and a conducting bundle. And we'll talk about those here in just a second, but let's start with those 2 nodes. So the 2 nodes are going to be the SA node, or the sinoatrial node, and the AV node. Now, the AV node is the atrioventricular node. Do make a note that both of these nodes, the SA and the AV, are both located in the right atrium. So they are both going to be within that first chamber that blood is coming into from the body, the right atrium. So with the SA node, make a little note here that it is going to be called the pacemaker. So super important that it is called the pacemaker, and that is because it has specialized cardiac muscle cells that are going to generate these spontaneous action potentials. And then these action potentials are going to pass to the atrial muscles and then to the other node. Speaking of the other node, which is the AV node, it is going to produce action potentials much slower than in any other part of this particular system, and that's going to ensure that the ventricles receive a signal to contract after the atria have contracted. So if that gets thrown out of, I guess, pace, that's not going to be a good thing. Next in the conducting system is going to be basically like different pathways of how we are going to pass on the information from the nodes. So these are going to just be pathways through the heart to pass on that information. So the AV bundle is going to pass through a hole in the cardiac skeleton and kind of go through down the middle the interventricular septum. So kind of that little septum that's separating the ventricles. Our AV bundle is going to be passing right through the top of there. From there, it's going to split, and we have the right and left bundle branches. These are going to extend beneath the endocardium to pieces of the right and left ventricles. And then we have this term Purkinje fibers, which are going to be relatively large in diameter and have few myofibrils. They're going to have quite a few gap junctions and conduct action potentials to the ventricular muscle cells. And then here we're going to have a picture of kind of everything we were just talking about. So I'm going to kind of go through all these lovely things. And of course you can be my guest and read this, but I'm just going to go over kind of the anatomy of what we were just talking about. So the first is going to be this guy up here, that sinoatrial node, the SA node. And remember, it is going to be the pacemaker and send action potentials down to our AV node, the atrioventricular node. So again, both of those are going to be located within this right atrium up here. Now, this structure that I'm outlining is the interventricular septum, and as I said on that previous slide, this is where we're going to find our AV bundle, this guy up here. Remember, that's going to split, and that's going to be our right and left bundle branches. And then from there, we're going to go up and kind of around on both sides to those ventricles, and that's where we're going to have our Purkinje fibers. If you remember from the first semester of A&P, we define the membrane potential of a cell as the electrical charge difference across the plasma membrane. This charge difference is the result of a cell's regulation of ion movement into and out of the cell. Cardiac muscle cells, like other electrically excitable cells such as neurons and skeletal muscle fibers, also have a resting membrane potential. The membrane potential when the cell is relaxed. So just a little reminder of what that resting membrane potential is. The resting membrane potential depends on a low permeability, so how easily something can pass through the plasma membrane to sodium and calcium, and a higher permeability to potassium as well. So when neurons and skeletal muscle fibers and cardiac muscle cells are depolarized to their threshold level, that's when we're going to have an action potential. So just to make sure we're all on the same page, and I'm sure you guys know this, and I don't have to repeat, anything from your previous semester, but skeletal muscle is going to just be like any muscles that are voluntary. So the muscles that you can control, like your rectus abdominis, your gluteus maximus, your biceps brachii, triceps brachii, all those good things. So I'm sure that's bringing back a little bit of PTSD from learning all the muscles and their origin, insertion, and action and all that good stuff. But just to kind of keep in mind, that is what we're talking about when we're talking about skeletal muscle. So on this slide, it is talking about the differences between skeletal and cardiac muscle physiology, so their function. I could go through this and just actively just read off the slide, but I do want to keep this lecture a little bit on the shorter side, and there's still quite a bit to go. So if you want to pause it and kind of just read through, it should be a lot of terminology and things that you've already seen when it comes to the skeletal muscle tissue, but the new cardiac muscle physiology, it should be relatively self-explanatory in how they're different. But if you do have questions, please don't hesitate to ask. So this is what I wanted to get to. So I really like this little graph here showing skeletal versus cardiac muscles, the action potentials of both. So do make sure that you review the skeletal muscle and then take a peek at how different the cardiac muscle action potential is in comparison. So this is a really nice graphic. You can find it on page 693 in your textbook if that is something you want to look at. Otherwise, kind of read through. It's really self-explanatory here, and really interesting to see the difference in action potentials between skeletal and cardiac muscle. So just make sure that you're kind of reading through, and if you need to pause on this slide, go ahead and do so now. So the heart is said to be autorhythmic because that means that it is going to stimulate itself to contract at regular intervals. Kind of an interesting fact about the heart is if, unfortunately, it is removed from the body and maintained under physiological conditions with proper nutrients, of course, and temperature, it will continue to beat autorhythmically for a long time, which is really kind of a cool thing for heart transplants, if you think about it. So in the SA node, pacemaker cells generate action potentials spontaneously and at regular intervals. These action potentials spread through the conducting system of the heart to other cardiac muscle cells, which is going to cause voltage-gated calcium channels to open. As a result, action potentials are produced and the cardiac muscle cells are going to contract. I'm sorry, I didn't mean to say calcium just there. It's going to be sodium, guys. Sodium channels are going to open, and as a result, action potentials are produced and the cardiac muscle cells will thus contract. The depolarization of pacemaker cells is going to be dependent on sodium, potassium, and calcium. However, the way that these ions are going to affect the membrane potential are very different. When a spontaneously developing local potential, called the pacemaker potential, reaches threshold, then action potentials are going to be generated in the SA node. And then changes in ion movement into and out of the pacemaker cells are going to cause the pacemaker potential. And then here we do have that nice graph of the pacemaker potential going through those phases. So just take a peek at this. If you need to pause on this slide, go ahead and do that. Cardiac muscle, like skeletal muscle, which I know we've been talking about a lot, but it's kind of nice to kind of go back and re-talk about the things that you've already potentially talked about, have refractory periods associated with their action potentials. The refractory period can be subdivided into the absolute refractory period and the relative refractory period. So during the first, the absolute refractory period, the cardiac muscle cell is going to be completely insensitive to further stimulation. During the latter, the relative refractory period, the cell is going to be sensitive to stimulation, but a greater stimulation than normal is going to be required to cause an action potential. So because the plateau phase of the action potential in cardiac muscle delays repolarization to the resting membrane potential, the refractory period is going to be prolonged. The long refractory period ensures that contraction and most of relaxation are complete before another action potential can be initiated. This is going to prevent tetanic contractions in cardiac muscle and is responsible for rhythmic contractions. Now, if you've never heard of tetanic contractions before, just make a little note that it's just sustained contractions, and that word might look a little bit familiar. If you've ever heard of tetanus, that is what tetanus is basically causing. And keep in mind that tetanus is a bacteria that is going to cause muscle contractions and can end up stopping your breathing because it causes the diaphragm to contract, and thus breathing cannot occur. So, but obviously that's something else, but we're just talking about sustained contractions when we're talking about tetanic contractions. Next up, we are going to talk about ECGs, or electrocardiograms. So this is going to be a record of electrical events in the myocardium, and it can be correlated with mechanical events. So the P wave is going to be where we have the depolarization of the atrial myocardium and signals the onset of atrial contraction. And you can see that little guy here. Let me get out my little pointer. You can see that guy in green on my little diagram over here. He's going to be that first little blip that we see right, whoopsies, right here. If I can get it to draw for me. I can't, oh well. So next we are going to enter into the QRS complex. The QRS complex is going to be that big jump after doing a tiny dip, and it's going to end in that little dip. The QRS complex is going to be where we have ventricular depolarization and signals the onset of ventricular contraction, and then the repolarization of the atria simultaneously. So that's going to be that big guy right there. So this is telling you kind of like in general what's going on this little diagram. So kind of pay attention to what's in these parentheses. Next we have the T wave. The T wave is going to be this kind of longer, not quite a blip, kind of a little bit of a foothill, if you will. And this is going to be the repolarization of ventricles that is going to precede ventricular relaxation. And then again, in parentheses, it does tell you that there. There are a couple other terms I want you to kind of recognize here too. The first is the PQ interval, or PR interval. It is going to be 0.16 seconds. The atria are going to contract and then begin to relax. And then the ventricles are going to begin to contract, and the QT interval is going to be 0.36 seconds, and that's where the ventricles contract and then begin to relax. And that is what this bottom part is showing you down here. So the PQ, or the PR interval, and the from here to here, whoopsies, there we go, the QT interval and how long they last. So if you want to kind of think about it, all of this is happening so incredibly fast. I mean, can you even imagine, you know, just 0.16 seconds or 0.36 seconds? So in less than a second, both of these are, if I could draw properly a straight line, both of these are happening in less than a second, and that is, you know, essentially your heart beating, right? So all of these things are happening very, very quickly. Here we're going to have a few alterations that we can see in electrocardiograms. Some of these are kind of interesting, looking and reading electrocardiograms. The first is going to be a complete heart block. This is where our P waves and QRS complexes are not going to be coordinated. And you can see all those P waves kind of popping up. It's just completely uncoordinated, not a good thing. The next is going to be a premature ventricular contraction, or a PVC, and that means that there's no P waves that are preceding the PVCs. Next is a bundle branch block. and you can see those prolonged QRS complexes, and I know I'm kind of just reading off this, but I think they're kind of interesting because you can see how an electrocardiogram should look. So looking at these, you can really kind of figure out what's going on and why it's wrong, right? And then you can read the last two. I think the first three are kind of more interesting, but maybe you find other ones are interesting, but let's move on. All right, popping on into the cardiac cycle, the heart is two pumps that are going to work together. We kind of understand that by looking at the anatomy. We see that, obviously they are right and left halves, the right atrium and right ventricle, and the left atrium and the left ventricle. So two pumps working together to cause your heart to beat. So the cardiac cycle itself is just a repetitive pumping of those two halves of the heart in a process that begins with the onset of contraction and ends with the beginning of the next contraction. So when we're talking about these contractions, we also have to acknowledge that there are relaxation periods, and we're specifically talking about these chambers. And what we call those contractions and relaxations is going to be systole and diastole. So systole is going to be the contraction of a chamber, while diastole is going to be the relaxation of the chamber. And then blood is going to move through the circulatory system from areas of high to low pressure. Think of diffusion. So diffusion is going to be solutes and water and things of that nature, technically osmosis, but moving from areas of high to low. So we are going with the gradient. We're not going to ever go against the gradient. That would make life so much harder. We don't want to do that. We want to make this as easy of a process as possible. So high to low. Before the cardiac cycle begins, all chambers are going to be relaxed and blood is going to come flowing from the veins into the atria and passively into the ventricles. So everything's kind of open is how I want you to imagine that. Most of ventricular filling occurs during this time. When the atria contract, active ventricular filling is going to occur at the force of atrial contraction, which is going to essentially, if you want to think of it, kind of top off the ventricles. So imagine everything sort of like flowing in from the atria to the ventricles. That valve is open. The tricuspid and bicuspid valves are going to be open, and you just have this nice filling. Again, the ventricles are going to become mostly full, but then the atria are going to squeeze and push all the rest of the blood that's in the atria into the ventricles that were in the atria. And under most conditions, the atria are going to function primarily as reservoirs. And the ventricles can pump sufficient blood to maintain homeostasis, even if the atria do not contract at all. During exercise, however, the heart pumps 300 to 400% more blood than during rest. As heart rate increases during exercise, atrial contraction is important for ventricular filling because less time is available for passive ventricular filling. Therefore, it is during exercise that the pump action of the atria becomes important for maintaining the pump efficiency of the heart. During the previous ventricular diastole, the ventricles were filled with 120 to 130 milliliters of blood. The volume of blood in the ventricles at this point is the end diastolic volume, or EDV. As the ventricles begin to contract, ventricular pressure rapidly increases, resulting in the closure of the AV valves, so the bicuspid and tricuspid valves, right? Ventricular volume does not change during the period of isovolumetric contraction because all the heart valves are going to be closed. So real quick before we jump into the period of ejection here, I want to point out that the millimeters of mercury measurement that we can see on this slide here is just a unit of pressure equal to the pressure exerted by a column of mercury 1 millimeter high at 0 degrees Celsius is all it's really talking about. just kind of keep that in mind, that that's all that is referring to. I'm not going to really ask you about that, but if it was something that was kind of confusing, just kind of make a note of it at first. So as soon as ventricle pressures exceed the pressures in the aorta and pulmonary trunk, the semilunar valves are going to open. The aortic semilunar valve opens at approximately 80 millimeters of mercury ventricular pressure, whereas the pulmonary semilunar valves open at approximately 8. Although the pressures are different, both valves open at nearly the same time, so kind of make a little note of that. As blood flows from the ventricles during the periods of ejection, the left ventricular pressure continues to climb to approximately 120, and the right ventricular pressure increases to approximately 25. The larger left ventricle pressure causes blood to flow throughout the body, which is going to be systemic circulation, whereas the lower right ventricle pressure causes blood to flow through the lungs, which is going to be the pulmonary circulation. It is important to note that even though the pressure generated by the left ventricle is much higher than that of the right ventricle, the amount of blood pumped by each is roughly the same. So during the first part of ejection, blood flows rapidly out of the ventricles, and towards the end of ejection, ventricular pressure decreases due to reduced blood flow, despite continued ventricular contraction. By the end of ejection, the volume of blood in the ventricles has decreased to about 50 to 60 milliliters. And then the volume of blood remaining in the ventricles at the end of ventricular systole is going to be called end systolic volume, or ESV. Completion of the T wave results in ventricular repolarization and relaxation. The already decreasing ventricular pressures are going to fall very rapidly as the ventricles suddenly relax as ventricular diastole begins. When the ventricular pressures fall below the pressures in the aorta and pulmonary trunk, the recoil of the elastic arterial walls, which were stretched during the period of ejection, is going to force the blood to flow back toward the ventricles, thereby closing the semilunar valves. Ventricular volume does not change during the period of isovolumetric relaxation, and this is going to be because the heart valves are closed at that time. So those AV valves. The relaxed atria were filling with blood during ventricular systole and the period of isovolumetric relaxation. During ventricular diastole, as ventricular pressure drops below atrial pressure, the atrioventricular valves open and allow blood to flow from the filled atria into the ventricles. At this point, the atria and ventricles are going to be relaxed. Blood flow from the area of high pressure in the veins and atria toward the area of low pressure in the relaxed ventricles. But remember that most ventricular filling occurs during the first 1/3 of ventricular diastole. At the end of passive ventricular filling, the ventricles are going to be around 70% full. So everything that we've gone through kind of up to this point in the last, I think, 4 or 5 slides has been what occurs during the cardiac cycle. So you can go through that again. You can kind of read through these little summarized blurbs about the cardiac cycle using this really lovely and very well done illustration. But literally everything that we've just covered absolutely covers the events occurring during the cardiac cycle. But kind of go back through, make sure you kind of understand what all is going on, and maybe looking at this figure on page 699 in your textbook, or you can just have this slide open and re-listen to the last couple of slides here. Would probably help make it make sense. You know, illustrations do definitely help if you're a visual learner. If you have someone near you and they are, of course, a willing participant, I would highly suggest listening with your ear to their chest of course, over their heart, so slightly to the left of the sternum, and listen for these heart sounds. It's kind of cool to listen to these sounds as you are learning about what they are. So the pumping of the heart is going to produce two very distinct sounds, and this is going to be classified as lub and dup. So these sounds are best heard by applying a stethoscope at particular sites in relation to heart valves. And the first sound is going to be that lub. It is a low-pitched sound, and it occurs at the beginning of ventricular systole and is caused by vibrations of the atrioventricular valves and surrounding fluid as the valves snaps shut. The second heart sound is a higher pitched sound, often described as dup. It occurs at the beginning of ventricular diastole and results from the closure of the two semilunar valves, so the aortic and pulmonary valves. Systole is therefore approximately the time between the first and second heart sound. Diastole, which lasts somewhat longer, is approximately the time between the second heart sound and the next first heart sound. And then there is a third, which is occasionally heard, but typically it's going to be heard in those who are maybe slightly underweight or quite thin, as well as young. It is caused by blood flowing in a turbulent fashion into the ventricles, and it can be detected near the end of the first 1/3 of diastole during passive ventricular filling. So these are the spots on the chest, the thoracic cavity, where you have those valves located and where you would potentially want to be listening, where you're going to have the best chance of hearing the heart. Moving on into the aortic pressure curve, the elastic walls of the aorta are going to be stretched as blood is ejected into the aorta from the left ventricle. Aortic pressure is going to remain slightly below ventricular pressure during the period of ejection. And do remember that as blood leaves the ventricles, the pressure in the ventricles begins to decrease, even as the ventricles continue to contract. Similarly, pressure within the aorta will decrease as well. As ventricular pressure drops below that in the aorta, blood flows back towards the ventricle because of the elastic recoil of the aorta. As the blood flows back toward the left ventricle, the aortic semilunar valve is going to close. Pressure within the aorta will rise slightly. Then you're going to have a sudden change in aortic pressure, resulting in a dicrotic notch or excursia. This is going to be the aortic pressure curve. This term, dicrotic, means double beating. When increased pressure caused by recoil is large, a double pulse can be felt. Aortic pressure then gradually falls throughout the rest of ventricular diastole as blood flows through the peripheral vessels. When aortic pressure has fallen approximately 80, the ventricles again contract, forcing blood once more into the aorta. Many of us have had our blood pressure measured during a medical exam. I'm sure you have, using potentially the pressure cuff and someone listening at your elbow. Blood pressure measurements performed for clinical purposes are going to reflect the pressure changes that occur in the aorta rather than the left ventricle. The blood pressure in the aorta fluctuates between systolic pressure, which is about 120, and diastolic pressure, which is about 80 for the average young adult at rest. So those numbers should sound kind of familiar. We'll talk about that a little bit later, but just remember 120 and 80 is the numbers that we were just talking about. Here I want you to loosely understand just two equations. The first is going to be the mean arterial pressure, which is going to be a pressure that is just slightly less than average blood pressure in the aorta. And how we figure out the mean arterial pressure is going to be taking cardiac output and multiplying it by the peripheral resistance. So our cardiac output is the amount of blood pumped by the heart per minute. There is a little sub equation to figure out cardiac output, and that's just stroke volume, or the blood pumped during each heartbeat, multiplied by heart rate. So the number of times the heart beats per minute. That is going to be our cardiac output. The peripheral resistance is going to be the total resistance against which the blood must be pumped. And so that's how we figure out the mean arterial pressure. There is another little term here I do want you to know underneath the cardiac output bit there, and that is the cardiac reserve. This is going to be the difference between cardiac output at rest and your maximum cardiac output. Here we have a nice little chart showing us the factors that affect our mean arterial pressure, and you can look over this. Again, this lecture's getting a little long, so if you need to, you can pause it and take a peek. The last part of this lecture is going to talk about the regulation of the heart in respect to maintaining homeostasis. So in this case, we're going to talk about intrinsic and extrinsic regulation, which is going to be how the heart functions to essentially balance the needs of the body. So if your tissues are needing more oxygen, more nutrients, something like that, your heart is going to be responsible and responding to that need and do some type of output in order to meet those demands. So intrinsic regulation is going to be the result from normal heart function, while extrinsic regulation is going to involve neural or hormonal control. So let's talk about the intrinsic regulation first. So you have something called preload. The preload is going to be the amount of stretch of the ventricular walls of the heart, and the greater the stretch, or the preload, the greater the force of contraction is going to occur. And this, you can write down, is known as Starling's law of the heart. So it's just going to be what describes the relationship between the changes in the pumping effectiveness of the heart and changes in preload. Another intrinsic regulation is going to be the afterload. It is the pressure the contracting left ventricle must produce to overcome the pressure in the aorta and move blood into the aorta. The afterload can be related to the amount of force necessary to open a door, if you want to think of it as kind of like a real-life example, if you will. And then just make a note that the heart is not as sensitive to the afterload as it is to changes in the preload. Just make a little note of that. So these extrinsic regulations of the heart, again, are going to involve neural and hormonal control. The first we're going to talk about is the parasympathetic stimulation. This is going to be supplied by the vagus nerve. So make a little note there that it is the vagus nerve specifically that we were talking about here. That is going to decrease our heart rate and acetylcholine, our neurotransmitter there that we're going to be talking about, is secreted and hyperpolarizes the heart. The next is going to be the sympathetic stimulation, and this is going to be supplied by a different nerve, the cardiac nerves. This is going to innervate the SA and AV nodes, which again, remember, they're in the right atrium, coronary vessels, and the atrial and ventricular myocardium. It is going to increase the heart rate and force of contraction, and the two hormones, epinephrine and norepinephrine, are going to be released. It is also going to increase our heartbeat and cause an increased cardiac output. It's going to also cause an increased force of contraction and cause a lower in systolic volume. The heart is going to empty to a greater extent, and then the limitations of this is going to be that it has to have time to fill. And just to kind of continue on that last little bit, here is our hormonal control, epinephrine and norepinephrine from the adrenal medulla. Remember, the adrenal glands are going to be located on top of the kidneys, and this occurs in response to increased physical activity, also possible emotional excitement and stress. Continuing on with the heart and homeostasis, so maintaining that optimal state of existence is how I always like to think of homeostasis, kind of just existing, surviving, living. Let's talk about the effect of blood pressure. Baroreceptors are going to monitor our blood pressure. The term baroreceptor, not sure how much they went into it last semester, but I always like to think of baroreceptors as like if you think of like weather type stuff, barometric pressure, it's just talking about that pressure, right? In the walls of internal carotids and the aorta, this is the sensory information that is going to go to the cardioregulatory center in the medulla oblongata, so that part of the brain stem, which can then increase or decrease the heart rate. So we're just sending that information there to be processed and then to put out whatever we need to be done in order to fix whatever it was, in this case, in response to blood pressure, what needs to be done to help us get back into that optimal state, right? Moving on into the effects of pH, carbon dioxide, and oxygen. Chemoreceptors, as the name implies, are going to be receptors that are going to be picking up on chemicals, specifically that measure pH, so the acidity and basicity, as well as carbon dioxide levels found in the hypothalamus. Chemoreceptors are going to monitor your oxygen levels found in the aorta and internal carotids, and prolonged lowered oxygen levels are going to cause an increase in your heart rate, which is going to increase blood pressure and can thus deliver more oxygen to the various tissues that are going to be needing that oxygen. Here we have a term called chemoreceptor reflexes. And this is going to regulate the heart activity to maintain homeostasis of pH and blood gas values. So that's oxygen and carbon dioxide, what we're talking about specifically. So when we're talking about ions, essentially, typically the three big ones that we're always going to be talking about, especially when we're talking about something that involves, muscles and those various things, is going to include the ions, calcium, potassium, and sodium. In this case, we are going to be talking about the increase or decrease in extracellular potassium, causing the decrease of the heart rate, which can result in a heart block, which is going to be the loss of the action potential conduction through the heart. And then talking about kind of the effect in relation to homeostasis, of course, is what we're talking about, of body temperature, how that can affect the heart and its homeostasis. Heart rates will increase when body temperatures increase because that is going to affect your body's metabolism. As you increase temperature, most organisms in general, and humans are no exception, as you increase the body temperature, it's going to increase your metabolism, your respiration, your heart rate, etc. And as heart rate decreases, well, not as heart rate decreases, but as your body temperature decreases, as will your heart rate. So everything's getting colder, everything will get slower. Try to conserve its energy to those central areas. So again, temperature increase equals heart rate increase. Temperature decrease is going to cause heart rate decrease. And then here is just some of those reflexes, the baroreceptor and chemoreceptor reflexes, and if you'd like to read that, you definitely can. I'm not going to go into it in too much detail, so that's kind of up to you. We've already sort of talked about it. And then more of that, just a summary of the baroreceptor reflex. And then lastly, a summary of the chemoreceptor reflex. And that's all I've got. I am so sorry that this lecture got to be so long. Please make sure to get your quiz done. If you have any questions, please don't hesitate. Have an absolutely wonderful day. Make amazing choices. You're doing great. Keep doing you, being you. You are wonderful. You got this.